outside and got a great seminar today. Our own Professor Steve Benight. Dr. Benight got his bachelor's degree in physics at Pacific University and then became a Rambling Rec. Went to Georgia Tech, right, for master's and PhD, and did his postdoc at University of Washington. And then immediately moved to his first faculty role at University of Illinois, Chicago, and where he rose through the ranks quickly to full professor, and then moved in 2003 to PSU, where he's been ever since. He's been very productive. He's closing in on about 100 papers. He has 16 patents, and I know him as the go-to DNA person in my life, anyway. And he's done so many wonderful things. He's very expert business person, several startups, a lot of experience in the startup world. And I know when I have questions about that, I always seek his advice. And his work, very interesting, from all the way from plasma thermograms to binding and interactions between proteins and drugs, discover pharmaceuticals. So he's going to tell us, Dr. Benight today is going to tell us about his sabbatical, specifically discoveries and evolution of new technologies, the applications in drug discovery and development.
Well, thank you all for being here. It's a pleasure to see you, and it's hard to bring you in from the beautiful day outside, but we won't get sunburned in there, I promise. So, thank you for that warm introduction, Professor Strong. I really appreciate it. I'm glad to be here today to talk to you about what I did when I was gone a couple of terms. Some of you might have not noticed, but I was gone a couple of terms on sabbatical. And what a sabbatical is about is to go out and, first of all, do things that maybe you hadn't got done. Dot some I's, cross some T's, get some paperwork done, get some reporting done, whatever stuff that you just don't seem to get to when you're in a hectic life at the university teaching and curriculum and other types of things. So it's a good time to go out and just reflect. We should be able to have that. You can also have the freedom to travel in the middle of the week, or the schedule at the end of the term and so forth. So I took advantage of that, as you'll see. So this talk's going to take a couple of different parts. First part, I'm just going to do a map of where I traveled to and why. As Rob mentioned, I've started several companies, and I actually have another company founded now. We're going to talk about some of the core technology of that company. But part of a startup, you have to learn, and you try and fund your company through grants from the government because you're good at that if you're a professor. Ostensibly good at it if you're a professor. But you also have to find other sources of income, revenue, in order to fund the research that you're going to try and get over the top to make a product. So when I went into science, I knew I had the choice to choose the problems that I wanted to work on. And all the problems that I chose to work on were of a fundamental nature, as you'll see. But I always wanted to make sure that I had an end product or something at the end in mind that it could lead to, because I figured that was the way for paying everybody back, society, for funding my education for all those years and all letting me kind of live a very, comfortable lifestyle. And so I chose to make products, or try to make products. And you'll see some of those in the making here. I've made several, and Rob said I've had several companies that sold them. So that's where I am. And so I want to talk about first traveling around, and I was getting to investments. The really neat investments, of course, you have the grants. Then you have the venture capital, the vulture capitalist, and then you have, depending on your story. But there's really, the ones that really carry you across the canyon are the 3 F's. Friends, family, and fools. You want to get those people to invest in you, and they have to sort of, at some point, either have a huge appetite for risk or love you and don't want you to see, be sad about your company. But anyway, I'm going to talk most of my traveling was involved traveling with my family and my wife. size family, but in that circle of people, there's some family that are potential investors. Of course, I wasn't shy about telling you about the opportunity. And so I want to talk about that. And then the next step, I'm going to talk about the basis, the sort of scientific basis of the technologies that I'm going to describe. I'll talk a little bit about the data, review sort of the literature that we've created. And then I want to talk about a very, very exciting discovery made by Matt Eskew in my lab about 4 years ago. And that is, and just digest this, I want to put it in your head so you can think about what it might mean. I have some suggestions, but we discovered that the heat capacity and the transition temperature of a melting of a protein, which you measure in a calorimeter, and I'll talk about that, and DNA are the same per mass. What's the heat capacity? Well, it's what influences. Well, it's the what is the definition of the amount of heat required to raise a substance 1 degree, 1 degree C or something like that, so it depends on the substance, and there's a constant in there called the specific heat. So anyway, you measure this, and the experiments that we found that at the peak height of the transition, in both variety of proteins and DNA, the heat capacities are the same per mass. And that's just the result of making sense. So we'll talk about why, and then at the end we'll talk about applications of that discovery, and then we'll talk about a theoretical explanation of what the observation means that makes those two things different. So I'll try not to go over. I've got slides prepared till 6:30. I have some sandwiches coming in about an hour. I'll try and finish in record time here. So anyway, this is a picture of Big Sur, Central California coast, where I live part-time with my wife. This is the famous Bixby Bridge that you see there, that you always see on maybe the sitcom Sweet with Big Little Lies or something like that. It's the Pacific Road, that's where we live. And so I thought this picture was a perfect backdrop because I admit that I did spend last winter down there instead of here. So the title really is, the top part is sort of buried in the clouds for a reason. Fundamental thermodynamic studies of proteins and ligand binding. Okay, so we do some science and we learn some stuff, and then we see if we discover anything in that process, and through that process, this technology evolved. That is, can you see a potential market for it? And what are the applications where that market's going to serve? And this is about applications in drug discovery, development, and screening. So now we're going to go start on my travels. Here's a map of that country. But the first stop we made was to visit our collaborator, LSR Barber, who's the chairman of the Department of Biophysics and Chemistry at Oregon State. She's a Portland State PhD. She graduated from her colleague David Payton, mentored her back in the late 80s. So she's risen to really some high repute and fame. She's an NMR expert, mainly, as you could guess, working for Dr. Payton. But she's also works on a very interesting field of protein classes that aren't included in the Nobel Prize that was just awarded 3 weeks ago or so. They're called intrinsically disordered proteins. So they have an ordered part and a disordered part. And they're involved in a variety of diseases. One of them is P53, an anti-tumor gene that you've probably heard of. It has a disordered, highly disordered domain and an ordered domain. So we're going to talk, she supplied samples and was a collaborator on several grant proposals, but that's LSR. So Rob mentioned I did a postdoc at the University of Washington, and I worked for this guy who's now retired. His name is J. Michael Shearer, Mickey Shearer. He's a lot like me, kind of, or excuse me, misspoke there. I'm a lot like him in that he's a physicist that's been working on biological molecules in the chemistry department his whole career. So that's sort of like me, a little bit. I'm a physicist, but I've worked on biology in the chemistry department as far as research for a career. So anyway, the point is to show Mickey up here. I have to go up and sort of like just meet with him and check in because he's kind of my barometer and whether my head is going crazy with the things I was thinking about, particularly in the theoretical side. And he had a lot of input on the heat capacity equivalence that we're talking about. Yeah, this here is my brother Barry. Barry is a professional engineer. He used to live 32 years in the Silicon Valley. He graduated from college in 1982 and grew up in Silicon Valley, worked at Stanford and where he got his undergraduate degree. Went to Stanford and then worked, and when he got out, he worked at Stanford for a while, and then he became just one of these engineers in Silicon Valley where you go to work for a nice fat salary, you don't like it, or your boss don't like you, you walk across the street and get a raise. So you just hop around and work in 30 different companies in the Silicon Valley. Why did I mention that? Well, he's an advisor. He's my family too. He's not much of an investor, he's really tight. But he is an advisor. And he's worked with me in the past on a number of my companies, computer-related computational things. His latest gig when he retired was he worked in Google Research, pre-AI, which was public, so he was all into that years ago. He got fed up with the San Jose area after living there for 40 years and moved to Dallas, Texas about 4 years ago. And that's him at his farm. He's got a horse farm and a horse ranch. It's a farm with no horses, but he's got about, I don't know, 7 or 8 cattle that he's messing with. So that's Barry, and we rely on him for expertise in the computer world. This is my daughter, her husband Zach, and my grandson Octavian. They live in San Mateo, California. My daughter is also a PhD from the University of Washington, and she went to Stanford, but she also has her own business now, and she's very active at a company in Seattle called Nonlinear Materials. And they gave a seminar, both of these people, a couple of years ago. The idea is that you get organic molecules and you design them properly and you know what you're looking for, and you get them to orient and produce nonlinear effects. What's the point of that? Well, the idea of that is that you can push a current along with photons instead of electrons. So it's called the R33. There's this funny nonlinear effect. If you shoot the light in this way, the signal comes up that way, highly coherent and highly pure. So her company has been involved in sort of doing that and commercializing that. The point is that she has extensive expertise in business development in the Bay Area. So I'm relying on her for that. Zach has started several biotech companies. He's in his 3rd company now. They just raised, I don't know, some kind of money, I think $12 million in the Silicon Valley for a trade startup. So I visit them quite often, but I also have a plan to get my grandson there, one of his trains. Okay, this is a wedding that we went to. My wife's nephew got married. These are all part family but part investors. The guy in the foreground there is Jeff McKee. He's a lawyer in Phoenix, and that's James' brother. And then the guy standing up closest to us is a guy named Trip Hurd. Very amazing guy, retired cardiologist and radiologist, also a submarine sailor. So he was in the nuclear power program for 8 years. He's a potential investor and a real physics nut. The guy sitting down there is Carl Rando. He's a very impressive guy. He's an old family friend. I didn't know this. But Carl's a securities lawyer from Phoenix. Actually, he's from Detroit originally. Securities lawyer. And Carl controlled and helped people invest money in the Phoenix area. A lot of rich people in Phoenix and all over the world. He is in charge or knows of a fund. His funds are funds that can invest, make investments that a bunch of people throw their money in. Not like your mutual funds almost. And make interest and make investments. He controls a fund that he told me is nearly evaluated at nearly $1 trillion. So there has to be some kind of sovereign wealth, and we figured either Saudi Arabia or Norway. Those are the only countries that have that much sovereign wealth. Okay, so anyway, they're all, and then his son there is Phil, what's his name? Oh, Hankey. Phil Hankey. He's an investor from Texas too. They're all potential investors. This is my cousin Shane Dixon up in the upper left-hand corner. We moved to Little Rock, Arkansas. I was born there, and my parents met there. The guy in the upper left-hand corner is Shane Dixon. He owns a trucking company in Conway, Arkansas. He's related to me, thank God. He runs a company that's nationwide. He only has 190 trucks on the road at any one time. The picture to the right is me and my wife in front of the house that my grandmother lived in, that my mother was raised in Dartnell, Arkansas. right across the river from where I was born in Russellville. They met at this place here in the lower left-hand corner at Arkansas Tech University, like the Georgia Institute of Technology. Arkansas Tech University, they met, and I was born there a couple of years later. And here finally is I think our last stop. Matt and I went to a meeting in Washington, D.C., where he was invited to selected to give, it's called the Defense Connect, where all the big DoD contractors get together and you can make pitches to them. Well, this pitch won the finals, and we got the pitch to, we don't know, but all the people in it, the big guys that control the first strings of the military in that talk did a great job in pursuing those opportunities. So that's where I traveled, 6 or 7 stops, just so you know. Did business all the way, worked on papers and other stuff. But mostly just trying to talk to these people all over the world. So the overview of my talk, first of all, I'm going to talk about the main instrument, the main technique we use. It's differential scanning calorimetry. I'll describe that. Second thing is, we're going to use differential scanning calorimetry for, well, we're going to do thermal denaturation studies of proteins. What's that mean? We're going to melt them. We're going to heat them up, watch them come apart, and measure the heat changes involved in that process. While you're raising your temperature at a linear rate, what's going on with the molecules is also reflected in the heat changes involved in the conformational changes that they do. And if they bound by something, that also affects the heat. That signal is very powerful in what we've capitalized on for our studies. Rob mentioned the plasma thermogram. About 15 years ago, got involved with some people at the University of, well, I've known them for years, but they were at the University of Louisville Cancer Center. They had the crazy notion, the calorimetry experts had the crazy notion of putting plasma into a calorimeter. So I'll talk about the data that you get from that. The plasma thermogram is indicative that differential scanning calorimetry might be a diagnostic, but there's some very big hurdles to overcome. Turns out the plasma thermogram is a melting curve of plasma. It contains about 6,000 proteins, but only about 9 of them are prevalent enough to predict the DSC signal when they melt. So we'll do a list of those, and that one major component that's about 50 to 65% of plasma is human serum albumin. So the rest of the majority of our studies now from here will be done on human serum albumin. We'll use calorimetry for analysis of the ligand binding and complex media. This is a big challenge because a lot of drugs are produced in vivo in tissue culture or some kind of culture, and then in some kind of big lysate. So either by bacteria or human cells, and there's a big lysate, and they're trying to overproduce a protein that they've just made a genetic construct for, and they don't know how much they've got. In order to find out how much they've got to have enough to survive the 25 isolation steps and 4 days it takes to figure it out. Big bottleneck in drug discovery. So what we were able to show, Matt was able to show, is that we can analyze ligand binding to specific protein targets in complex media, and even more than that, evaluate the binding constants. Also, the technique we're able to show that you get quantitative determination of ligand binding constants and binding stoichiometry. This is a big question. It's something you know it binds. You want it to have some kind of effect in the clinic. What's the stoichiometry? What's the stoichiometry? Is it tetramer, dimer, octamer, whatever? So the technique can also show you how to do that. And then the big coup de grace here or whatever is the calorimetric determination of protein mass in pure or complex mixtures. This is the big deal because you might have an unknown protein that's being expressed that you want to see if it's in there. But all you've done is you see a little peak. It's giving you a rise that there's a protein there. What you can do is you measure that peak on a differential scanning calorimeter, get a value, and then because the DNA and the protein peak capacities are the same, we can make a standard curve of concentration in the DNA. It's constructed from the calorimeter melting curves I'll show you, and then you can just look down and figure out whatever that peak height is, reflected from the process tells you the concentration, the unknown concentration in the protein. So I'll show you that, and that's pretty cool. Preclinical drug discovery applications, and then finally, the discovery, which I just have been talking about for quite a while now, the discovery is that the heat capacity equivalents, and I'll talk a little bit about some results of some theoretical work with some group I've been working with, the theoretical physicists from Russia. I've been working with them for about the last 15 years. They finally came up with a model, statistical mechanically based, that shows that there's one of the reasons why the heat capacities are the same is that because there's hydrogen bonding that can occur within a partially denatured structure. This is a whole different phase that occurs that people haven't appreciated yet. Everybody knows water's involved somehow, protein stability, but they don't really know how. People say there's structured water, water's ordered, and all that. Well, in this case, it's true that water gets ordered, but the solvent doesn't order the molecule orders the water. Because there are only a few of them, orientations, as we'll see, that combine with a partially denatured structure. Okay, here's the machine. Very simple. It's a differential scanning calorimeter. Plug it in. I think this one here was about $110,000. Pretty expensive, but it's slow. You can only do, just one sample at a time. But the principle of the technique is really, I'm sure y'all know, I'll just review it for you. On the left is a sort of the sample cell. It's a bunch of coils that will heat the sample in the reference. In the middle is a schematic of this, and the whole idea is this. You have a sample and some buffer, you have that buffer. And what you do is when you heat them both up at a constant rate, linear rate, with an ambient heater, shown up there at the ambient heater over here. You see, is that showing up? Anyway, there's the ambient heater upper left-hand corner. And so what happens then, as you heat this thing up, and the sample has molecules in it, they undergo some kind of a transformation, melting, conformational change. That's reflected in the heat change in that particular subreaction. And then what happens is, that heat is then compensated for the reference. What does it take for that? Say it's an exothermic reaction. And it's going to have more heat than the reference had. So what the instrument tells you is, now I got to provide more heat to the reference to make it equal to what the sample is. And that's the differential scanning, differential part of the calorimetry, because that heat that you have to apply to get the reference back up to what the sample is, since I have an exothermic reaction going on, that's proportional to the enthalpy, which is a fundamental quantity in thermodynamics, but also the basic parameter that we measure. So you get essentially the power, the raw sense, the power you have to supply. As you look at this, as you heat this sample up, to make the reference equivalent to it, and then it's already heating up to the differential heat involved in that process is why it's called differential scanning calorimeter, because the difference between the two. All right, here's what it looks like. This is just a simple diagram of what the experiment, the data looks like. You get a curve that looks sort of like that one on the right. The excess heat capacity, which you measure in the calorimeter, versus temperature. And on the low temperature side, the protein now is denaturing, melting. But the way that it works, the peak, and this is a really very intriguing part. The peak is a very interesting part of this whole transition that is not well understood. I think it's a transition state, a phase transition. And I'm not ready to say that to you because you will write it down if you want to try to publish it. I think it's a phase transition. So what happens is you go up to the peak as you put the sample up, 50% of the molecules melt. And exactly at the peak, which is called the TM, there's an equal population of both melted, denatured molecules, and intact molecules. And so it doesn't really matter how you do the statistics. There could be a mixture that there's an equal amount of unbound in one and bound in the same one, likewise. But the point is that half of them are still intact. As you increase the temperature further, you go past the TM, then what happens is the fraction folded goes down, because what you're looking at up there is most of them are already denatured, and then what you're going to be denaturing is the rest of the ones that are still folded. So what that is a measure of essentially that. As you get to the bottom, then you basically blow everything apart. But for proteins, that's a problem because they like to aggregate, particularly human serum albumin. You know, albumin is the major component of eggs. What happens when you heat an egg up? It turns into jello, you know, really viscous stuff. And that's when, if you put too much human serum albumin in a differential scanning calorimeter, you will clog the cell. And if you clog the cell, it's a week to clean it out. So, you don't want to put too much in there. First thing I do is say, "Phil, it's too big." Don't do this as a function of concentration. How high do I go? It doesn't matter. But then I go in and plug the machine until they'll learn how to clean it out. Sorry, manipulation. Anyway, it's good for it. This is the plasma protein. If you take plasma, which is the fluid that comes out of your blood, it's not a red blood cell, not a white blood cell, but includes the clotting factors, fibrinogen. Serum is everything in plasma except for fibrinogen, the clotting factors. So if you take this isolate of blood, you can isolate it out of whole blood. If you find that it's comprised of a number of proteins, and these proteins can be drug targets, they can be single molecules, just everything. It's just a plethora. The proteome is really very rich. So try to understand, since we're going to put plasma in a calorimeter, you gotta have to have appreciation of what the composition is. I know these did this by myself. This is a group effort with the University of Louisville. And the point Brad shares there. The point is that albumin is the largest component by far comprising the plasma proteome. And so generally, if the hypothesis is, well, if the thermogram of the plasma is perturbed sufficiently, more than likely not that some of that perturbation is coming from something that's binding the albumin in the plasma. So that was the albumin hypothesis. So here's the human plasma thermogram on the left. You can see it's a little different than the simple melting curve I displayed before, single peak. It's got two peaks, but in the middle, the first peak, this peak here, this peak here, it has mostly HSA and mostly haptomomen. Remember in the previous slide, HSA was about 55%, so that's most of it. And of course, you got the immunoglobulins down here. There's transferrin out here. There's fibrinogen. It's not there when you don't serve. Okay, so you see this, you're going, wow, that's a pretty. The information technologists that I've talked to when they approach about this, the machine learning guys, and these guys can find anything. So there's not enough signal there. They're used to looking at mass spec, it's got all these spikes, and that's a lot of information. They just view all this information. There's not a whole lot of information here. So we had a little bit of a challenge in trying to make it diagnostic. So what we did is got a bunch of samples from the University of Oklahoma Lupus Repository. Plasma samples, blood samples. The Lupus Repository, these were a cohort of patients that were family related. So we took sort of that genetic variation between them, but they were matched. Like one sibling has lupus, the other one doesn't, that kind of thing. And there were over 1,000 of these samples that were examined from all demographies, all racial, men, women, black, white, Hispanic, old, young, everything. And what we found is that pretty much most of the time there were variations in sort of the normal pattern, which is this one here, right? This one here is just like the normal pattern. But there was all this stuff over here. We didn't understand what it was, and we still don't understand what it was, but the correlation was those were Hispanics, male or female. For some reason, the thermogram was shifted. I don't know what it means. We always thought maybe it was hot sauce or something. But no, seriously, it was a quandary, especially to the immunologists that we talked to, the people, the rheumatologists that were interested in this, because there's some kind of lupus is some kind of autoimmune disease, right? So something there, I still don't know the answer to that. The point is that there's a difference here, in different groups. If there's a difference between different groups, there should be a difference between, and this is the same disease, there should be also differences, you would think, within the sort of standard one that are irrelevant. At least some kind of chemical diagnosis. So we had to look at more than one. We went up to Toronto and consulted with Dr. Martin Frisler, who was kind of like the world lupus expert. And and mostly for autoimmune diseases. We supplied a number of samples for him. He's done a pretty cool job. His job was, he's an MD. His job was just to supply as much of these kind of samples, these disease samples for these autoimmune diseases. It's good because they're often misdiagnosed with one another, and they respond to treatment very differently. So here's the thermograms for normal is the black, myositis, rheumatoid arthritis, lupus, and scleroderma. These are all autoimmune diseases that are misdiagnosed, also with Lyme disease. And so, if you have Lyme disease, you want an antibiotic. You don't need some kind of other drug that they give you. So what you see here on the right is they're all broken out, and those are at least 25 samples. No, at least 25 samples of each of these. So you see up in the left-hand corner, you see normals with myositis, terrible autoimmune disease. In the right corner, you see rheumatoid arthritis. In the left-hand corner, you've got lupus, and you've got scleroderma. You can see that for each one of these disease states, the thermogram appears to be different. Well, they go, voila, it is a diagnostic. It is sensitive enough to see differences. Well, you have to do a lot of studies, and we did a lot of studies. We did a study with the Mayo Clinic. We did an extensive study at the Brown Cancer Center. And the motivation for doing all of this is that once we saw the difference, we started looking at the literature. We never looked at that before we started the experiment, we were biased. So anyway, we looked at the literature. There was over 40 publications in the last decade. We attempted to use DSC as a diagnostic tool based on the kind of observations that we made. It turned out, though, in all the publications we saw that the utility of the DSC was limited greatly because they didn't really know what was causing the thermogram perturbation. Okay, so myositis thermogram shifts, rheumatoid arthritis thermogram shifts. What causes it? Well, you can think of a number of things. Maybe remember, this is like mostly albumin. Maybe in the disease state, the albumin, or maybe you have myositis, the albumin is bound by something. Or maybe that state makes an abnormal albumin, which isn't likely, because without normal albumin, you're not going to be around long. It transports a lot of stuff in the blood. So you don't think it's too much of a mutation in the albumin. The logical one is, okay, it's something that's being secreted, obviously, okay, a ligand being secreted with this binding with the albumin. That's what's causing maybe the thermogram. So consequently, because of those inabilities, we can't make any differential diagnosis because we don't know what it is. So the way that the whole field, before we sort of jumped into this about 7 or 8 years ago, was about 9 years ago now, is that people were taking these thermograms and they were trying to tease out detail and use sophisticated statistical routines to see if there were small changes that they could make a big deal out of. Well, I don't like that. I like big changes that you have to try to make a small deal out of. So it turns out that the statistics, don't get me wrong, anybody that does it for a living, you gotta be careful. And it's just like Rutherford said, he discovered the nucleus of the atom, key to atomic theory, atomic structure. If your experiment needs statistics, you have to have done a better experiment. There's a lot to be said about that. Except like everything in those sections. Okay, I'm going to talk about some results now. We're just going to fly through these, and I'll try and summarize what each one of these manuscripts are showing us says. This is sort of just a summary. I'm doing this for 2 reasons. One, to show off a little bit, not kidding. The second is to give you some background to where we're going. So you see, this is how we do this, because I'm going to state some things that maybe I have to go a bit about. Okay, so the results. The first thing, we said we gotta see if we can get anything out of plasma. Anything in plasma. So we said, well, albumin's in plasma, and we should be able to, if the ligand's bound, binds albumin, it ought to be in the plasma. So we ought to be able to take the whole solution of plasma and fish out the albumin-bound ligands by modifying something that binds albumin, streptavidin and biotin, a couple that used to make these assays. And so you make an albumin that has a biotin on it, and the biotin then you use to throw it in the plasma, and then it binds the that can retrieve the ligand that is needed to be put in there, see if you retrieve it. So you throw in something in the plasma, throw the albumin-bound albumin in there, and see if you can capture what you threw in. When we did that, did mass spec on it, showed that we could retrieve a ligand that we put into the plasma by just building a capture reagent that was only based on the albumin. So the albumin was modified. Of course, if you modify the albumin, you've got to prove that you didn't change the albumin. That's the next step. Anyway, this was the first patent on this stuff that we've been out about 4 or 5 years now, 4 years ago. But it was the capture reagent for a strategy for retrieving these ligands from plasma. People that tech transfer all say, oh, this stuff's all been done before. You just pull down the assays, everybody's done it. I said, please, just call the patent attorneys and let me talk to them. You gotta call the patent attorneys and they say, oh yeah, this is very proprietary. I don't know what that means. Anyway, the second one is ligand binding naturally modified serum albumin. Since we made the capture reagent, so if we're gonna use this, we gotta make sure we're not messing with the albumin. Okay, so the way we modified it was biotinylated albumin, and published a paper on that. And that we're able to detect the binding constant and read it analytically. Next is build binding constants for human serum albumin enhanced by ratiometric analysis. This is a blood down. You have the albumin, and then you have the ligand. And what you do is you have the albumin alone, then you throw it, and then you melt that. Then you throw in the ligand, and then you melt the albumin in the presence of the ligand, and from the shift that you get, the differential you get, or the ratio of the shift you get due to the binding of the ligand, tells you you're able to evaluate a binding constant. How you do that? Well, you do biochemistry. What do you do? Well, you titrate in whatever you're binding to as a function of concentration, stay below the binding constant, and fit the curve with it, right? Okay. Next one is the equivalence that I talked about. Equivalence of the transition heat capacities, and I'll talk in detail about this. So since I've mentioned it several times, I'm going to skim over this, at least the summary. Then another one is finally thermal analysis of protein stability and ligand binding in complex media. This is what I talked about a little while ago. You have an expression system that you're creating a protein. You want to see if you're trying to maybe screen drugs against that protein. So you want to throw in the drug, and you want to find out how well it binds. Well, you have to be able to do that to save time. Like I said, you'd like to be able to do that in complex media, which is either tissue culture lysate or bacterial lysate. And we did that. That was some years ago. And then finally, the patent, and this is the basis of our SBIR grant. This is the DNA-based assay for calorimeter determinant protein concentration in pure mixed solutions, which I talked about. I outlined a couple of times. Like I said, I'm going to show you specific examples of that in just a second. This was also a patent that the university had no problem going to patent it, because this is a monumental discovery. The implications are very big. And so to make it a technology which the NIH agreed is really a novel approach. Okay, so now let's talk about some specific results of some specific experiments. This is HSA drug binding. We took the ratiometric approach that I'm talking about. We looked at a variety of drugs to see if this was just one-off or did it work for a lot of drugs where the data, the binding constant for albumin was in the literature. So what we did is look at 29 different drug ligands that were tested in all this variety of different drugs. Also, I'll say that what came out of this work, the drug doesn't necessarily have to be water soluble. We developed a way to solubilize a non-water soluble drug and then use it in a calorimeter in a aqueous solution. And then we could also evaluate the binding stoichiometry. So we'll first talk about the ligand thermodynamics. Let's go right over this real quick. The idea is we want to measure from the measurements that we get when we determine the free energy for binding. This is dependent upon the binding constant or the dissociation constant, Kd, 1 over Kd. The point is that you get a thermodynamic profile. Thermodynamic profile different processes that can contribute to the calorimeter. And overall, we should use the free energy, but here's a component for the internal structure. Here's a component for the conformational change. Here's one for desolvation as a function of temperature. So all of these processes, chemical processes, are captured in the thermogram. What you do is you just integrate the thermogram, and the area under that curve is the inflow. The area under that curve divided by T, integrate that curve is the enthalpy. Excuse me, control. Okay, well when you bind a protein, it's all about the Chavier's principle. You throw something in, something changes because of what you threw in. There's something the chemistry related to what that thing you threw in did. The fact that it could change the position to establish a new equilibrium in a solution with no drug. You throw a drug in, the equilibrium is going to redistribute in that mixture. That's the Chavier's principle. And so what we observed universally is that when ligand binds to HSA, it increases the stability. And that increase in stability is a function of how much drug you throw in there, how much ligand you throw in there. So from that, you're able to take the signal that you get from the DSC curve, the response to that event, you can then plot that versus the concentration of what you threw in. Now we see this is the case for albumin in the presence of increasing amounts of naproxen on the left, increasing amounts of BCG, both produced in green. There are two generic ligands, cheapest in the Sigma catalog. Naproxen, we know, is an NSAID. I think the trade name is Elite, I believe. I'm not sure. And so what you measure here is the calorimetry experiment is the heat capacity on the left is a function of temperature for naproxen. And those are increasing amounts of naproxen. You can see that there's a titratable increase, shift to the right in the presence of both of these drugs. These shifts aren't exactly the same because the mode of binding is different for these two ligands. Naproxen binds differently to procreosol than procreosol 3. And we can detect that as well. So what you do is you take that signal and you create the sort of classical dose response curve. You use two parameters from the thermogram, the free energy, which I talked about, and then the transition temperature, the TM. So the peak height shifts lower, and then the free energy we calculate as a result of the collective integration of the curve itself. So what you see here, in the proximate for TM, this is this one here, we get a binding constant of 2.2 times 2.2 micromolar. For free energy, it's 2.1 micromolar, so it's fairly close. Here, over here for BCG, actually it's a little, quite a bit smaller, in fact, 20 times smaller. This here, though, is also different. So you get a different result for the TM than you do for the free energy as a function of now the concentration. So what that tells us, something that we discovered again was, wow, that ratio of what the TM analysis divided by the free energy analysis, the KD you get from that gives you the stoichiometry of the binding. We said, oh, that can't be. So what is the stoichiometry? The known stoichiometry of naproxen and BCG for HSA. Wow, 10.8 to 3.0. That's what we evaluated. So anyway, that's another example. Okay, here's another example. We then looked in the literature and said, no, there's like all these drugs that bind HSA, and they're published, the binding constants are published. How well does our technique work? So we went out and we did the experiments, did all these DSC experiments and all these different ligands. But the point is, this is the result. Ours are in red, the published are in black. Just about in every case, I think save one, we are dead in the error of binding. So our technique really works. It works as good as anything else that's out there. Okay, now we move to complex fluids. We'll talk just a little bit the process of constructing a complex fluid, at least what we're talking about. We make a gene, it goes in an organism, it can be a tissue culture cell or it can be a bacteria, but you make some kind of gene construct. And that gene codes for some protein that you hope you can get overexpressed or get expressed so you can harvest it from the meat. So what you do is you transfect the cell with the construct, it gets incorporated into the organism DNA, then the cells proliferate, then what they produce protein, and then protein you want here, in this case, is the little green ones. Then you lyse the cell, and then what you see is that now you've got this emission stuff that you got from lysing the cell, and you want to extract these through this green one here, which is the protein of interest. And so the challenge is to go from here to here, like I said, is 4 days and 5 days and 27 steps, 4 or 5 columns, no pain in the butt. So anyway, the idea, if we just want to determine how much protein is being made in the cell lysate, here's an example. We're looking for this protein which is overexpressed. This is a gel electrophoresis pattern, and this is just a gel where you load molecules on statement, and all the different molecules under electric field migrate to a different part of the gel, different distance in the gel. So what you see here, we have a bunch of smear. This is a molecular weight marker. These are the smear. Here's the first lysate. Here's the second one. Here we go. Now we're starting to purify it. Here's some other stages of purification. What you can see here as you move across, the other bands disappear and the bright one gets brighter. That means you're getting close to purified product. That takes a lot of time. And if you're screening 1,000 compounds in a week, you know, or creating trying to grow up 1,000 different constructs a week, you want to assay for the protein you're making, that can be a real limiting factor. So it's really cool if you can determine the binding constant to whatever drug you're screening to the protein that you're making in the complex fluid. Here's an example of a complex fluid. On the left we have human kidney cells, the black, that's just the lysate when this construct was made. Then the black is the cell lysate with the expressed protein, which is angiotensin converting enzyme, an isoform of that. You might have heard of it because people that have high blood pressure often take a ACT inhibitor, because what it does is it binds to the surface cell. What happens in the surface cell is it also does something with the calcium, the sodium channel. So they have inhibitors of ACT, that lowers your blood pressure. Also, it turns out there's a lot of range studying ACT with the COVID outbreak, because apparently the same entry to the cell, at least for the receptor binding domain of COVID-19, is also binds to the same site as ACT. There's a lot of interest in that. The point is here, we got this from the collaborator with an overexpression lysate. Here's the lysate by itself. Then we've extracted the background out of this to get this black curve. You see we've got this sort of Humpty Bump camelback up here. This is purified ACT that was also supplied by the collaborator. You'll notice that there's 2 peaks here in the unpurified. There's only one here in the purified. Okay, why is that? Well, it turns out ACT is comprised of 2 domains, a C domain and an N domain. And oftentimes, they're both required for proper function. And it looks like the purification got rid of the N domain. This peak is no longer here, right? It's here, but the purified doesn't have a peak there. Okay? So it looks like purification somehow eliminated the N domain. Well, our collaborator was very interested in this. He said, well, that really makes sense. We looked at the literature, and lo and behold, we found a study where they take an ACT and they remove the carboxyl domain. And what we saw is that this is the carboxyl domain, the N domain, C domain, N domain, and it turns out that peak goes away. Here's the ATC, it's very similar to ours. This peak goes away. So that kind of proved that the purification had basically destroyed the N domain of the protein. Okay, now a bit of a home stretch here. I want to talk about Matt Eskew. Matt Eskew, we worked together. He was my graduate student for 5 or 6 years. We've been working together about 8 or 9 years. We have a lot in common and a lot different. We have in common that he grew up in Chicago and I was there for 17 years. So we sort of got that same mindset. That helps. You know what it's like to get up real early before it's light and get to work and not get home till it's dark, because that's what you do in Chicago. Anyway, talking about that work ethic here, I'm glad he did, because here he is. He got his PhD thesis in 2021. I wouldn't allow him to get a haircut in until I finished writing his thesis. But essentially, with a tome, it's sort of a story of algebra. In fact, we're writing a book based on his thesis. The thermodynamics of liquid binding and local structural stability of human serum albumin. And so, it weighed over 3.5 pounds. Okay, Matt discovered the equivalence of heat capacity, as I indicated. For globular proteins, it had already been in the literature that at 25 degrees for several globular proteins, the heat capacity there, so the very before the thermogram even starts, the heat capacity there was approximately the same. They all had about the same heat capacity, which was the total area, essentially. Transition heat capacity of the protein is defined as the maximum peak height. So we define the maximum peak height as different from the heat capacity at 25 degrees, but the heat capacity is measured over the whole curve. This is the one at the TM, the transition temperature. We define that as the transition heat capacity. Taking the thermogram peak heights, this is how you analyze those, and I'm not going to go through any more than just to show you. You can show that the maximum peak height is made up of a term due to the protein, and then other terms due to the buffer and reference and machine using. The point is, it's proportional to the mass. This is the mass, this is the density times the volume. When you boil this all down, make some assumptions, do the experiments on this thing in a systematic manner, and what you get is an expression from the maximum heat capacity is this very simple linear equation. That is that the heat capacity of the protein minus the partial specific volume of the protein. That's just the volume that the protein molecule itself excludes. Okay, so that's the partial specific volume. And this is the times the density. What that means is that what I'm meant to show here, is that this is a linear equation. It is linear. The way you can determine from the slope, you plot the maximum peak height versus the mass and the concentration. And what you should, the slope of that curve, if it's linear, should be directly the heat capacity of the protein minus the partial specific volume of the protein. Okay, so here's a plot, two different globular proteins, HSA, lysozyme, multiple experiments. Those look pretty linear. This is basically the maximum peak height versus the protein concentration in both cases. And so they're pretty linear. Remember that I said that the slope of those curves in that linear equation is just the protein, the heat capacity of the protein per mass minus the partial specific volume. Turns out also, what's remarkable about this story is that these proteins have very different partial specific volumes. 0.733 for HSA and 0.003 for lysozyme. That's a big deal for volume. Okay, so there's the equation that we fit the line to evaluate the protein heat capacity. Just take the slope. And what we did is in both cases, lo and behold, if you look at the heat capacity, the transition heat capacity for both lysozyme and human serum albumin, it's basically the same per mass. 2.86 microcals per gram per degree Kelvin in both cases. Increase in the second decimal point. Okay, well, okay, boy, that could just be fortuitous. That's only two proteins. Well, what about DNA? Get the wild idea. Let's see if DNA does this. So as Rob mentioned a long time ago, we did a lot of DNA work. So I worked on DNA hairprints. These are these molecules that can form on themselves, have a self-complementary sequence. Here's an example of a sequence that does that. So we did the same business on this. We melted as a function of concentration. Lo and behold, you can take those same equations and just plug DNA for protein and evaluate the DNA, maximum DNA specific heat capacity. So it has a much different partial specific volume, 0.55. We get almost exactly the same heat capacity per gram. For DNA, remember it's 2.82 for lysozyme, 2.82 for HSA. Now for DNA, it's 2.86 plus or minus 0.03. So DNA and protein have the same heat capacity. Even though they have different partial specific volumes, they have different stabilities. So we just couldn't take enough of this and put another DNA, one that melted at a 20-degree higher transition temperature. So do the same thing? Surely this different structure is going to change the heat capacity. did it. Here's the linear plot. Here's the fit. Again, 0.55 is the partial sitting volume. 2.87. We've got 2.85 for the DNA that's 20 degrees less stable. Wow, that's a big deal. I mean, I didn't know what I was doing. I thought I got some paint there or something. It's odd. It's amazing. It's truly amazing. So, here's a summary of all those things. If you look at the high temperature hairpin, the low temperature hairpin, here are the transition temperatures, vary over a factor of 30 degrees. Here are the heat capacities per gram. Well, this is for DNA and this is for protein, so per residue value, if you take per base pair, or per base, or per amino acid, they're different. The transition heat capacity for mass is exactly the same. Well, you said, okay, got to be something in there. Maybe it's the salt. We melted these all in PBS milk from 4.1 or sodium chloride. So we changed them all over and put them in cesium chloride. Cesium chloride is way bigger than sodium, so we said maybe this is some kind of electrostatic effect in the solvent. Well, people say this is in experiments got exactly the same heat capacity in all those conditions. Holy moly, what is going on here? So it occurred to us at this point that we could use this fairly robust deal, the quality is a fairly robust deal. We could use that in order to develop a technique to determine unknown concentrations, as I alluded to before, from the DNA curve. We know the DNA concentrations. We make a standard curve, just like you do here, and then you use this to determine the unknown concentration. How do you do that? Well, here's the protein and DNA equations, basically the same one, the slope plus the PSV, same thing for DNA. You can equate all these, equate the DNA, these two things for the protein and for the DNA, you can equate those. When you equate those, you solve for the density of the mass, which is the volume. So you can determine the mass of the protein just by the maximum peak height of the protein and the corresponding concentration on the standard curve for the DNA, minus the difference in the partial volumes of the two molecules. Here's another example of that, and that's the standard curve. Well, did this for a bunch of different proteins, so that's really cool, but does it really work? And the answer is remarkably well. Here's a bunch of different proteins, HSA, lysozyme, SARS-CoV-2 receptor binding domain, ACE2, HSA and plasma, E. coli, lysate, ACE2 unfiltered, streptavidin, and 16 were linear DNA. Well, we took the big guys, here's the partial split bonds and vary all over the place. Here's the assay determined mass. Here's what independently determined by different means. So there's good agreement all the way across the board. Furthermore, for streptavidin in the 16-mer DNA, which is, we noticed that there's a factor of almost 4 difference between what's independently determined and what our method showed. It's a factor of 3.95. Likewise, for linear DNA, this is without the loops, same sequence without the loops, there's a difference of 1.81, almost 2. Well, streptavidin is known to be a tetramer. Duplex DNA is a dimer. So our method determines the monomer concentration. Okay, what good is any of this? We'll go back to LSR, my very first slide. LSR provided us with a protein that is a tertiary disordered protein, the LPD. It has ligands or cofactors that bind to it. She wanted to know if she's looking for drugs, right, that can perturb the protein. So we could bind the curve. She supplied these ligands, these OD and OTQ and everything. Anyway, we measured, from our technique, we measured the first measured the thermograms and then determined the concentrations of the different molecules, these 3 different ligands essentially, without a ligand and then the disordered domain by itself, and then with the 2 ligands. And what you find, we were able to evaluate this very complex system that they could not. We got they had spent years determining these binding, not years, but certainly a graduate student complained it took them 2 years to do it. And basically what you see is that we're very close with our method for this complex fluid, and more importantly, we were able to show that this disordered protein predicted something that they're still trying to measure. So And finally, here's a summary, just basically meeting accuracies and procedure, all under 10%, except for one, nutritionally disordered proteins, which is the industry standard. We felt pretty good about that, a lot of samples. Unfortunately, we tried to get a phase 2 grant, NIH grant for this, and the NIH wasn't convinced. So that was not funded, unfortunately, which happens again. We didn't get any unfunded, anyway, point is, okay, I'm gonna go through this real quickly. What this is there's a model based on statistical mechanics. I'm just gonna tell you the result because I'm running over time. First of all, I've been involved with these theoretical physicists in Armenia and Russia, and basically they've developed a model that was developed to look at magnetization in spin glasses. These things where the magnetic moments are the different components, and the lattice can be magnetized, and we'd like to be able to understand the distributions because why? Because you're going to make some high-tech material to run through it. So there's a number of these theoretical papers that we worked on. And here's another method, but there's about 5 or 6 of these. But finally, what they did, Artem was a graduate student at the time. I worked with his bosses originally. His graduate student came up with a method called differential scanning calorimetry of proteins in the so-called Zen-Bragg model. Well, I don't want to go through the Zen-Bragg model. It's from 1959, but what it essentially says is that units of a protein can be considered as units that are hydrogen bonded. Each one of those units, along with the discrete alpha helix, can exist in 1 or 2 states. They can be hydrogen bonded, or they can be broken, and then it also takes into account what's next to them. Is what next to them broken, or is what next to them hydrogen bonded? It has to go from broken to hydrogen bonded. That costs you some entropy reduction. There's a parameter that accounts for that. The point is you take the array, all the possible arrays of all the intact and broken hydrogen bonds in the protein, the sum over those, determine something we use in statistical mechanics to determine the thermodynamic parameters like the heat capacity. It's called the partition function. You calculate the partition function. Here's the model. Well, here's the result. In the old-fashioned Zen-Bragg model, where you just had either hydrogen bonded with the linear unit or non-hydrogen bonded, a very simple expression. It's just the equilibrium constant for breaking that unit, the bonds in that unit. If you include now, when these things break, they can bind to solvent water in an ordered manner, not just like bulk solvent, but they have a specific orientation. There's only a few of them that they can bind. It's called averaging over the water degrees of freedom when you make the calculation. So if you limit the number of conformations, just being everything like bulk solvent, to specific orientations that can form these hydrogen bonds. You get a parameter that's called S that depends on the temperature, the hydrogen bonds in the structure, hydrogen bonds with solvent, and then these symmetry terms for the water and for the neighboring pairs. The point is, you go through this, and trust me, this calculation is not totally straightforward. But essentially, you evaluate expression for the heat capacity. Exactly what we measure. Now we're looking, damn, any of these 3 terms here, can we make any supposition? Can we get the mass, only the mass dependence out of here? And you can. If you look at the middle term, it's 2n times the hydrogen bonding for the solvent, and just exponential with the solvent again, the symmetry term for the water, temperature at the Tm. At the Tm theta, fraction of intact bonds is 0.5, that's a constant. So that just turns into 0.5. These terms drop out because theta is constant at the Tm again, all the hallmarks of the phase transition. What you see here is the F. There's a term that depends only on mass, and that's n. Number of units in the molecule. So what does this say? It says that there's apparently a very important state for proteins where the water is interdigitated or interbound with unfolded residues of different degrees of unfolding. That's what it suggests. So from that you say, well, wouldn't that be a network of some sort? Precisely. Only made of water. The solvent is ordering the water. Thank you for listening to me. Here's the theory of what produces the nice thermal rate perfect agreement. That's all there is. Thank you very much for your attention.